This lecture talks about some of the chemistry that you will need to know to understand physiology. All chemicals are made up of matter, which is anything that occupies space and has mass. Mass is a measure of how much matter there is. Usually the units are expressed in grams or kilograms, and there are about 92 different kinds of natural matter called elements. Total number of elements on the periodic table is actually 120. Okay, but everything after the first 92 are elements that have been made by humans. On this slide, we have some atoms that are commonly present in organic molecules. This is a table 2.1 from your book. And the point that I wanted to make is that elements are the basic building blocks of matter. And the major elements are hydrogen, carbon, nitrogen, oxygen. Sulfur is also listed on this table. But that's not to say that others are not crucial in the body, such as calcium, which muscles could not contract without, and sodium for sending action potentials, and all kinds of things, and, for example, the chloride ion. But this particular table lists the atomic numbers and atomic masses of these 5 elements, and you should know this information. An atom is the smallest division of an element that has all the chemical and physical properties of that element. So you should realize that an element has a nucleus, and the nucleus contains protons and neutrons. Hydrogen, which is shown in this picture, only has a proton in its nucleus. It does not have any neutrons in its nucleus. And the electrons are in a cloud, which are shells surrounding the nucleus. You should know that the carbon nucleus has 6 protons and 6 neutrons, and the shells surrounding the carbon nucleus hold 6 electrons. Hopefully you remember that the protons are positively charged, the electrons are negatively charged, and neutrons are neutral. And the overall charge on an atom is then neutral. The protons and electrons balance each other out. The atomic weight is the number of protons and the number of neutrons. Electrons have no mass. Think of them like feathers. If you put a feather on top of a ton of bricks, it would not change the weight of that ton. So the atomic weight of carbon is 12. The atomic number is the number of protons in the nucleus, and the atomic number of hydrogen is 1. And the atomic number of carbon is 6. Once the atomic number changes, the atom changes. So atoms are known by their atomic numbers. What you can see in this slide is the electrons are in orbitals around the nucleus, and the lowest energy shell holds 2 electrons, and each shell thereafter holds 8 electrons. This is known as the octet rule. that atoms are stable when there are 8 electrons in their outer shells, at least the atoms that we will be talking about, which make up living organisms. If an atom has an unfilled outer shell, then that atom participates in bonding, okay, because it is trying to achieve the octet rule. The electrons in outer shells are called valence electrons. So these are the driving force for bond formation. An isotope is an atom that has a different number of neutrons from protons. So remember that the number of protons is the atomic number of an atom. If that changes, then the identity of the atom changes. But if the number of neutrons changes, then that is considered to be an isotope of the atom. For example, the mass of hydrogen is 1. It has 1 proton, that a positive charge. But the overall mass that you can see on the periodic table is 1.0078. That's because that is the mass if you add up and average all of the isotopes. So deuterium has a mass of 1 proton and 1 neutron, which is 2. Or tritium has a mass of 3, and so we write each 3. So deuterium and tritium are isotopes of hydrogen. Some isotopes are radioactive. This is because their unstable nucleus gives off energy. This slide looks at covalent bonds. Covalent bonds are formed when electrons are shared. Here you have hydrogen gas, H2, and 2 hydrogens come together and they are sharing the electrons. That gives them a stable outer shell. Remember that the innermost shell is stable with just 2 electrons. So hydrogen must pair up with another hydrogen in order to achieve stability. Methane and ammonia are also formed by covalent bonding. And what this slide is showing is 3 different modes of writing these molecules, these structural formulas. So you have the letters and lines, and you have the model showing the orbitals, and you have that's this one, which is showing methane, and then you have the space-filling 3-dimensional model, and you have the same thing for ammonia on this side of the slide. Some molecules form or atoms form polar covalent bonds. This is when there's an unequal sharing of electrons between 2 atoms in a covalent bond. If one of the atoms is oxygen, oxygen is electronegative. That means that it attracts electrons. And when oxygen bonds with hydrogen, it pulls the negative charge towards it, leaving the positively charged hydrogen. Another electronegative atom is nitrogen. Also, fluorine and phosphate are electronegative atoms. Polar molecules make great solvents. For example, sodium chloride or sodium chloride table salt will dissolve very well in water because of the polar nature of water. But let's look at ionic bonds. Ionic bonds are formed when one atom donates an electron to another atom. And of course, the purpose of donating an electron is to achieve a stable outer shell following the octet rule. So in this slide, you can see that sodium is donating its electron to chlorine and thereby forming sodium chloride, an ionic compound. But let's look at how sodium chloride dissolves in water. Okay, so what happens when you put sodium chloride into water is that, being it's an ionic compound, it's going to easily dissociate or separate. So in water, sodium chloride becomes the sodium ion and the chloride ion. And you can see that the positively charged sodium here is attracted to the negatively charged oxygens on water. And the chloride ion on the other side over here is attracted to the positively charged hydrogens on water. And then the interesting thing about what happens when water molecules surround these ions is that more water molecules are attracted to the first layer of water molecules, and these things called hydration spheres are formed. It's these hydration spheres that make a molecule soluble in water. The ability to make hydrogen spheres indicates that a molecule is polar, and polar solutes are charged, which makes them hydrophilic. So the polarity is due to uneven electron distribution due to electronegative atoms. Also, polarity is due to the asymmetry in the shape of a chemical. Just its geometric shape is asymmetrical. And also, lots of hydroxyl groups in a molecule will make it polar. Okay, because the hydroxyl groups, OH groups, are attracted to water. Ionic solvents are really polarity taken to an extreme because we actually have two charged ions. And then you have nonpolar solvents, which are like fats, which are hydrophobic or water-fearing. And fats float on water. For example, like oil and vinegar salad dressing with oil floating on the top. Okay, well, let's take a look at hydrogen bonds on the next slide. Okay, so hydrogen bonds are caused by an attraction between the hydrogen on one water molecule and an oxygen on another water molecule. So it's key that you realize it's the oxygen on another water molecule, because the oxygen and hydrogen within the water molecule is a polar covalent bond, but the hydrogen bond is formed between water molecules. Hydrogen bonds are going to form between any of the electronegative elements and hydrogen. For example, they hold the DNA strands together. And so in the bases that make up DNA, it's usually hydrogen bonding to nitrogen. And so what happens in water is, because it's a polar molecule, the hydrogen is more positively charged, and the oxygen on another water molecule is very negatively charged. And so you get this hydrogen bond, which is actually just a weak interaction between two atoms. Okay, so what do you think? Are these molecules polar or not? Water, methane, amino acids. There's a picture of an amino acid there for you to analyze. Oils, glucose. There's glucose up in the right-hand corner. And steroids, which is this ring molecule, is a steroid. So what do you think? Are these molecules polar or not? So hopefully you got these answers correct. And glucose might surprise you, but It's these hydroxyl groups that make glucose polar. So polar molecules are going to be soluble in water. And of course, the amino acid has this hydroxyl group here. Steroids are considered to be in the lipid family and are nonpolar. Okay, let's talk about acids, bases, and the pH scale. So here we have pure water, which is ionizing to form equal amounts of a OH group and a hydrogen. This solution is considered to be neutral. In neutral solutions, the hydroxyl and the hydrogen are in a concentration of 10 to the minus 7th molar, or 7 places behind the 0 moles per liter. An acid will be any solution with a higher hydrogen ion concentration, and a base is a solution with a lower hydrogen ion concentration. A base removes hydrogen ions from the solution. For example, sodium hydroxide is a base. Let's look at the reaction here. Sodium hydroxide binds with, or reacts with hydrogen ion, and that will form sodium ion and water. Let's take a look at the pH scale. So pH means the power of hydrogen, and there is the pH scale from your book. So the pH scale indicates the concentration of hydrogen ions in solution. Technically, pH is the negative log of the hydrogen ion concentration. The hydrogen ion concentration is 10 to the minus 7th molar, and the negative log of 10 to the minus 7th is 7. Okay, so pH 7 indicates a solution that is 10 to the minus 7th molar in both hydrogen and hydroxyl ions. And you should realize that since the pH scale is based on powers of 10, each pH value on the scale is 10-fold different than the next. Okay, so pH 5 is 10 times more acid than pH 6. pH 5 is 100 times more acid than pH 7. Then we talk about weak acids and strong acids. Okay, so a strong acid is like hydrochloric acid, and that will dissociate into hydrogen ions and chloride ions. So what you have to look at to figure out what a strong acid is how many hydrogens you have, because remember, the more hydrogens, the more acidic a solution is. So a strong acid, such as hydrochloric acid, dissociates completely into its ions. But a weak acid is not going to dissociate completely. Okay, so here we have a carbonic acid, and it is going to dissociate somewhat into bicarbonate ion, and you can see there are just a couple of hydrogen ions in this solution. The pH of the human body needs to be maintained between a fairly narrow range of 7.35 and 7.45. Because of this, the body has buffers. So let's look at a buffer. A buffer is a chemical that minimizes changes in pH. So it reduces the excess acidity and alkalinity. Just take a look at this hypothetical example of hydrochloric acid, which is a strong acid around pH 1, which we just looked at. But it reacts with sodium bicarbonate, which is baking soda and known to be a buffer. And the reaction causes the formation of sodium chloride and carbonic acid, which is a weaker acid. So in this way, the sodium bicarbonate buffer, or sodium bicarbonate, is able to act as a buffer. carbonated drinks contain carbonic acid, which then dissociates into carbon dioxide and water. And that's where the fizz comes from in carbonated drinks, which then after the carbon dioxide is released, they become flat. In the real situation in the body, the pH is stabilized by bicarbonate ion, which reacts with excess protons forming carbonic acid. In other words, a strong acid, hydrogen ion, is made into a weak acid, and the proton is taken out of solution. Okay, so blood pH is maintained by the bicarbonate ion carbonic acid buffer pair. Let's say blood pH goes down if you're exercising due to lactic acid releasing protons. The bicarbonate ion can pick up those protons and carbonic acid then forms. On the other hand, let's say a situation occurs where blood pH goes up, like in excessive vomiting, when you may lose a lot of stomach acid. And so carbonic acid is then going to dissociate into the bicarbonate ion with the release of a proton. In this way, blood pH is maintained in that range of 7.35 to 7.45, and conditions like acidosis and alkalosis are then prevented. Let's switch gears just a little bit and talk about organic molecules. Okay, so organic molecules, such as the ones shown in this slide, are molecules that contain carbon and hydrogen. Okay, so they are hydrocarbons. And they form because carbon can bond to four other atoms, and therefore carbon forms chains and rings through either single or double bonds, and sometimes even triple bonds. Classes of organic compounds can be named according to their functional groups. So we're going to talk about functional groups on the next slide. But realize that, here you have a chain or a ring, as in cyclohexane, or you have the benzene ring here. All of these are organic molecules. So let's look at the functional groups. On this slide, you have the carbonyl group, C double bonded to O, and the hydroxyl group, which is OH, sulfhydryl, sulfur, which is found on the amino acid cysteine, an amino group, which is NH2, found on all amino acids, a carboxyl group, which is acidic, C double bonded to O, and then also bonded to a hydroxyl. And then you have the phosphate, and then based on these functional groups, there are these categories of organic molecules that you need to know. So when the carbonyl group is in the center of two carbons shown here, this is a ketone. And here you have an aldehyde, the carbon double bonded to an O, but then also singly bonded to a hydrogen. The acid we talked about with the carboxyl group, forming an organic acid, was also down here. And the OH is found on alcohols. It's also important that you understand stereoisomers. These are molecules that have the same atoms in the same sequence, but their atoms are arranged differently in three-dimensional space. So the example shown on this slide, the first one is looking at cis versus trans. Okay, so cis means same side, trans means opposite sides of the molecule. And the examples are cis-2-butene, and you can see the methyl groups are on the same side, the cis configuration. But in trans-2-butene, the methyl groups are in opposite side of the molecule. This is a trans configuration. Then there are the optical isomers called enantiomers that are mirror images of each other. Here you have the D-glyceraldehyde and L-glyceraldehyde. You know, these are mirror images of the other, kind of like left and right-handed gloves. But if the palms are facing in the same direction, they cannot be superimposed on each other. And so this is pretty important in pharmacology. This slide covers the four main classes of macromolecules. You have polymers, which are the four main classes of macromolecules, and each of these polymers is made up of a monomer. So poly means many units, and mono is a single unit. So for example, carbohydrates are made up of simple sugars known as monosaccharides. Proteins are made up of chains of amino acids. Lipids are made up of fatty acids and glycerol. Actually, the lipid category is anything that is nonpolar, which also includes steroids. But the main lipid in the body is triglyceride, which is made up of fatty acids and glycerol. And then nucleic acids, such as DNA and RNA, are made up of nucleotides. On the next slide, you have the carbohydrates. And their simple structure is CH2O, carbohydrate. That should be easy to remember. And they have CH and O in a 1 to 2 to 1 ratio. This is the body's quick energy source. It's a major form of energy, and carbohydrates can be found in rings and chains. Most of the simple carbohydrates in our body are hexoses, hex meaning 6, and OSE ending, meaning sugar. You should be able to draw a simple glucose molecule, but the other major common ones, C6H12O6 in the body, are fructose, shown down here at the bottom, and also galactose. Pentoses have 5 carbons. So complex carbohydrates are formed through dehydration synthesis. Here you can see 2 glucoses reacting with one another, forming maltose, which is a disaccharide, with the release of water. So just think about the term dehydration, forming a bond while losing water. And in the picture down below, glucose and fructose react with one another to form sucrose with the release of water, and sucrose is your common table sugar. In the opposite reaction, so let's say to break apart these molecules, is called hydrolysis. Hydrolysis is the breakdown of polymers by the addition of water, so it's the opposite of dehydration synthesis. Just think hydrolysis, and you have starch that is going to break down with the addition of water into 2 maltoses, the disaccharides, and then maltose can further break down by the addition of water, hydrolysis, into 2 glucoses. And the slide shows you where the water, OH and H, adds onto the products. Okay, so a polysaccharide is a polymer of a monosaccharide. Common example in the body is glycogen. Glycogen is stored in liver and muscles. It is made up of monomers of glucose specifically, and it is a branched molecule, as you can see here. It is sometimes called animal starch, even though glycogen is actually more branched than starch. Plants store starch as their energy reserve, the way animals store starch as their energy reserve. It's a good idea to take a look at these different branched polysaccharides because we'll be talking a lot about them during the course. So we already mentioned glycogen, animal starch, alpha-1,4 glycosidic bonds. I won't ask you that. Remember that it's highly branched though. And then here's starch from plants, which of course we have a lot of in our diet, also formed by alpha-1,4 glycosidic bonds. But like I said, I will not ask you that. Oops. And here is cellulose. Okay, so cellulose makes up plant cell walls, but we do not have the enzyme to digest it. Cows, on the other hand, do have the enzyme, and they eat lots of grass, and that enzyme is in one of their 4 stomachs. Okay, so we'll be talking about glycogen, we'll be talking about starch, and somewhat about cellulose, but you should also be aware of chitin. Okay, chitin is another polysaccharide which has linkages that are similar to cellulose, but it has nitrogen in it. Okay, and the nitrogens make this molecule into poly-N-acetylglucosamine. And it's just interesting to note that chitin makes up the exoskeletons of arthropods. Okay, let's take a look at this student activity. See if we can answer these questions. Hopefully they're super easy for you. Which is a polysaccharide? Which is a monosaccharide? Okay, monosaccharide, polysaccharide. What is the ratio of carbon to hydrogen to oxygen in a carbohydrate molecule? Okay, so you should picture this, CH2O. So 2 hydrogens for every carbon and oxygen. Let's switch gears and talk about lipids. So lipids are all insoluble in polar solvents. Anything that's placed into the lipid category has been placed there because it is nonpolar. And at the top of the slide, we see palmitic acid, a saturated fatty acid. Saturated fats are bad for the heart. They build up in blood vessels with cholesterol, causing plaques. You know a saturated fat in your kitchen because it is solid at room temperature, such as what happens to the bacon grease after it cools down. Fat, lard, butter. They're called saturated because every carbon is saturated with hydrogens. All of the carbons are singly bonded to one another. So realize that a fatty acid is a very long chain of hydrocarbons with an acid group at the end. Okay, and then you have the unsaturated fatty acids, such as linolenic acid, shown at the bottom of the slide. Okay, and in unsaturated fatty acids, there are double bonds between one or more of the carbons in the hydrocarbon chain. This causes a kink in the molecule and makes it into a liquid called an oil. This particular molecule is an omega-3 fatty acid. That says that at least one of the double bonds is at the third carbon from the methyl group end of the molecule. And omega-3 fatty acids have been found to be beneficial to us because they are able to help to reduce cardiovascular disease. And here's some sources of omega-3 fatty acids: salmon, nuts, olive oil, and also avocados. Let's look at cis versus trans fats. So we already talked about the cis configuration versus the trans configuration. In oleic acid, the hydrogens are on the same side, cis configuration, around the double bond. But in allylic acid, the hydrogens are in the trans configuration at the double bond. Trans configuration being opposite sides. And you have probably heard that trans fats are not good for you. They are straight, so the double bond does not cause the kink in the molecule because the hydrogens are in the trans configuration. And so these trans fats can lay pretty flat next to each other, forming a solid. They have been found to raise total blood cholesterol, raise LDLs, low density lipoproteins, which are your bad cholesterol. They contribute to atherosclerosis, which is buildup of plaques in the arteries. And they lower the good cholesterol, or the high density lipoproteins, which actually help to prevent atherosclerosis, but the trans fats lower those. So want to stay away from things that say partially hydrogenated soybean oil, or fast food restaurants that heat and reheat the oil throughout the day. That has been shown to convert cis fatty acids into trans fatty acids. Let's look at the formation of triglycerides on the next slide. Just as we saw for complex carbohydrates, triglycerides are formed by dehydration synthesis. In other words, the removal of water. So you have a glycerol and 3 fatty acids shown here. And together they become a triglyceride. Triglycerides, I mentioned, are the most common lipid. I'm going to ask you to draw that, but you can just draw this simplified triglyceride, glycerol, and fatty acid 1, 2, and 3. When fatty acids are needed for energy in the body, triglycerides from adipose tissue can be broken down into ketone bodies. And here you have acetoacetic acid being converted to acetone. And so these are ketone bodies. They are 4 carbon compounds that are usually relied upon during fasting because the person is not taking in glucose and carbohydrates, and so the fat starts to break down. And often you can smell acetone on a person's breath as acetoacetic acid breaks down into acetone. And so that is a sign that somebody is fasting. Acetone breath would be due to ketosis, which is the breakdown of fats into ketone bodies. This may also occur in diabetics because they can't get glucose into their cells to break down glucose, and so they start breaking down fats as an energy source. The only problem there is that ketosis can result in ketoacidosis, which seriously lowers blood pH, and that could be deadly if blood pH is to go down. But ketone bodies are 4-carbon molecules that are the breakdown products of fatty acids. And the next slide is going to look at phospholipids. So phospholipids make up cell membranes. And here you have a central glycerol. And from 2 of the carbons of glycerol, there are fatty acid chains bonded. But to one of the carbons, there is a phosphate and also a nitrogen-containing choline group. Here is an artistic rendition of a phospholipid. Many artists draw it with a ball and 2 fatty acids. The phosphate is polar, or a hydrophilic, water-loving side of the phospholipid, and will be facing outward, outside the cell, and also to the inside of the cell in a lipid bilayer. And the fatty acids face to the center of the lipid bilayer. Fatty acids are nonpolar, hydrophobic, or water-fearing. And remember, like dissolves in like, so the fatty acid part of the phospholipid turns inward in the lipid bilayer. These molecules are sometimes called amphipathic. In other words, they are soluble in 2 conditions, both in fat and in water. Bile acids are also known to be amphipathic because they are soluble in both polar and nonpolar solvents. They're actually made up of cholesterol. And so let's look at what happens if you put phospholipids into water. These micelles form. Okay, and so bile acids form similar micelles in the small intestine. And these micelles help to break up fats that are coming through from the diet. The amphipathic nature of phospholipids also allows them to be used as surfactants. Okay, so we'll hear when we talk about the respiratory system that the surfactant on the inside of the air sacs prevents the lungs from collapsing due to surface tension that would be created if the layer inside the lungs was simply polar. Here's a simplified drawing of a phospholipid. We already talked about that, but there it is again. This slide is about steroids, which are nonpolar. And there's a picture of a steroid. The body converts cholesterol into steroid hormones, shown in this slide. Cholesterol and cortisol, testosterone, and estradiol. So cortisol is a stress hormone, testosterone is the male hormone, estradiol is the female hormone. Many of these hormones, testosterone and estrogen, is derived from cholesterol, and cortisol is derived from cholesterol in the adrenal cortex. These steroid hormones also play an important role in vitamin D formation. Another mini quiz for you. Can you draw the basic structures of steroids, phospholipids, and triglycerides? Okay, see if you can sketch that out. And I'm going to give you the answer right away. So if you're going to sketch it, you're going to have to stop the tape. Okay, steroids, phospholipids, and triglycerides. Then you have the prostaglandins, which have this pentose ring and fatty acid tails. These are regulatory molecules that regulate things like blood vessel diameter, ovulation, uterine contractions during labor, inflammation, blood clotting. Their name is derived from prostate, where they were originally found in the prostate gland. Let's look at some of these amino acids shown on this slide. Amino acids make up proteins, and there are 20 amino acids. They all have this general structure, which you need to know, of a central carbon, an acid group, and an amino group. And the 20 amino acids all differ in their R group. Okay, here's valine, here's tyrosine. You do not need to know the individual amino acids, but it is useful to look at how these functional groups give the molecules different properties. A whole class of neurotransmitters resembles the amino acid tyrosine. And there are actually many amino acids that are derived, or many neurotransmitters that are derived from amino acids, and they are called monoamines because they have one amine group. In the diet, 8 amino acids are essential that you must take in every day, and 12 are non-essential. Amino acids make up proteins, and they do this by the formation of peptide bonds. How do they form? Once again, by dehydration synthesis. The same way that triglyceride forms from fatty acids and glycerol, the same way that polysaccharides form from monosaccharides is how proteins form from amino acids with the release of water. And here you can see a peptide bond. It forms between the acid of one amino acid and the amino group of the other amino acid. And the amino acid sequence of a protein is known as its primary structure. So this would be a tripeptide. Okay. And proteins have primary, secondary, tertiary, and quaternary structures. The primary structure is the sequence of amino acids. The secondary structure is due to weak hydrogen bonds, and that can cause 2 different types of protein. These are regions of proteins to form. One is called the beta-pleated sheet or beta sheet, and the other is an alpha helix. And both of these forms can be found within a protein. And then the tertiary protein structure is 3-dimensional folding due to the interaction of those R groups we just looked at on amino acids. And these are affected by denaturation, which is due to heating, change in the protein structure due to heat and pH. Not all proteins have quaternary structure, only those that consist of more than one polypeptide chain, because quaternary structure refers to the interaction between 2 different polypeptides that make up the protein. One that comes to mind is hemoglobin, which has 2 alpha globin and 2 beta globin polypeptides. Let's also take a look at the tertiary structure here, and you can look at the different bonds that are responsible for the structure. Like here's an ionic bond between an amino group of one amino acid and a carboxyl group of another, or a hydrogen bond, or van der Waals forces, which occur between two very lipophilic amino acid side chains. Or a disulfide bond can form between the two side chains of the amino acid cysteine. And the tertiary structure is the one that we said could be denatured by pH or temperature extremes. This destruction is called denaturation, and is the same thing that you see in a fried egg. So you see the egg white lysozyme, and it was kind of gooey and clear when the egg was raw, but then becomes white upon heating. Okay, so let's talk about nucleic acids really quickly, such as DNA. Nucleic acids are made up of nucleotides, which have this general structure that you need to know. A 5-carbon sugar, a base, and a phosphate group. Together, when the linkages between the carbon of the 5-carbon sugar and the phosphate bond together, this forms a phosphate sugar backbone that makes up the backbone of a DNA sequence. The bases of DNA are guanine, designated by G, thymine, T, cytosine, C, and adenine, A. And DNA can form a double helix due to base pairing rules. C bonds with G, and the base pairs are made by hydrogen bonds, and A bonds with T. These are the base pairing rules. And when the hydrogen bonds form, here you have a G hydrogen bonding with a C, and down here you have the T hydrogen bonding with the A. This is what creates the double helix, which is shown on the next slide. And so here you can see this hydrogen bonding between the base pairs according to the base pairing rules, and the hydrogen bonding of the nitrogenous bases forms the ladder that you can see on the left-hand side of the slide, the rungs of the ladder. The sides of the ladder are formed by this sugar phosphate backbone that we looked at. Let's compare RNA to DNA. So RNA is ribonucleic acid, and DNA is deoxyribonucleic acid. And RNA is transcribed from the DNA code. And this is the first step in the expression of a gene. And RNA uses the adenine, guanine, cytosine, but instead of thymine, it uses uracil. The sugar is not deoxyribose, but is ribose. DNA, on the other hand, uses A, G, C, and T, and deoxyribose sugar. What's the difference between the two sugars? It has to do with the number 2 carbon on the pentose ring. In deoxyribonucleic acid, the number 2 carbon has a hydrogen, and in RNA, the number 2 carbon has the hydroxyl. So the oxygen is missing in DNA, hence the term deoxy. RNA tends to be single-stranded, but it can fold back on itself, creating double-stranded regions. It's also shorter than DNA in general. And so here you have RNA single-stranded and DNA double-stranded. DNA is longer than RNA and is a double-stranded molecule. Let's look at the different types of RNA that are found in the cell. 4 types of RNA are used to take information from the nucleus to the cytoplasm for assembling a protein, and also to assemble the protein, so taking the information out and then participating in assembly. The pre-mRNA, which is formed directly from a gene. RNA is transcribed from DNA. The messenger RNA, which is formed after the messenger pre-mRNA is spliced, and then tRNA, I guess this should be 3, tRNA, which is in the cytosol and carries amino acids to the ribosome for assembly into a protein. And we'll go back over that. And then ribosomal RNA, which is a structural RNA that is part of the ribosome. Some other types of RNA molecules also serve important functions in the body. Okay, either in energy or as second messengers in regulation or as coenzymes. Okay, ATP and GTP are energy carriers. ATP is the universal energy carrier made in the mitochondria. And then there's something called cyclic AMP, which is a second messenger and forms inside the cell in response to activities outside the cell. And when cyclic AMP forms, it triggers a lot of intracellular functions. NAD and FAD are energy carriers. They are coenzymes in that they help enzymes to function. They're actually derivatives of the B vitamins. And we will talk more about them when we discuss cellular respiration. But that is the end of our lecture on chemistry.